One thing, when you say that microorganisms are growing, what we actually mean is not that they are increasing in size relatively, it's that they are increasing in number. So microbial growth actually centers on the microorganism increasing in number, not increasing in size. All right. Now, they do like a kind of splitting, and then that makes them to increase. So if you're looking at a plate, now a dish on the microscope, and then we say that the microorganism is growing, what we are saying in a sense is that it's increasing in number. All right. So that's the first thing I want you to know. And of course, when we talk about the growth is all about the nutrition rather, it's all about resources that they need to grow. Now, one of the way we differentiate some bacteria is the kind of environment they can grow in. Right, so I'll be talking about different ones. We call some allophiles because of the pH factor. We call some thermophiles because they can exist in high pH, in high temperature. We call some of them generally extremophiles because they can exist in extreme conditions. Yeah, so generally I'll be looking at those things. One of the way we isolate bacteria as well is by putting them in an area when you know that it's only this kind of bacteria that can grow in that medium. So we call their nutrition, we call it medium. So you say, oh, can you put the bacteria in this media, agar media, all of that. Now that's what we use, and you hear it in the lab as well. Now, so when you see them, there are some times that a bacteria can be different kind of bacteria can be put in a medium or in a media, in a medium more appropriately. Then the ones that dies, the one that can survive, that the medium nourishes, will be the one that will survive in that medium. All other ones will not be able to survive. It's not because the medium is killing them, it's because they don't have nutrients to live on, so they cannot grow. All right. So those are one of the ways. So sometimes if you have a strain of bacteria and you want to isolate a particular one, that's something to do. You can put a colony in a medium. When you put it in a medium, you're going to notice that the one, when it's cultured, the one that knows, the one that survives in that environment, that is actually for that environment, will grow, while the ones that are not able to feed with that will not grow. All right, so that's one of the ways to isolate bacteria. So let's go. Now, I'm aware that I've not, okay, I'll talk about that then. Now, so microbial growth is increase in number, like I said. It's not the issue of size, it's increase, and it's because the microorganism, they will divide, they will go through reproduction process. Now, organisms that have, so result of microbial growth, they will start forming colonies. They will start, colonies means that they are gathered together. So you see clusters all around. Now, there is something we also call biofilm. Now, biofilm, it's more like this. It's a film. And when you say film, we are talking about something that laminates a surface in that way. So, excuse me. So you see bacteria more like it's laminating surfaces. And so we call that biofilm. 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 So it's, again, it's when you see bacteria forming a collection, laminating a surface. It's more like that. So you see the bacteria with matrix, glowing, being this, right? Now, organisms use a variety of nutrients for their energy, like I noted there, needs, and to build organic molecules and cellular structures. That's what I was noting earlier, that most bacteria, they have the ideal media for them, or ideal medium where they grow. And in that medium, there will be nutrients that will foster their growth such that they grow, increase, and they also build the necessary structures that they need. Now, every media will have carbon in them. They will have oxygen, nitrogen, and hydrogen that are essential for growth. Now, microorganisms could get nutrients from different sources, but the actual resource is their medium. Now, organisms that make their food, just the same way we always talk about it, are the autotrophs. Autotrophs make their food. The concept of autotrophs is that they are self-feeders. Self-feeders, autotrophs. Now, heterotrophs, they don't make their food. That's fine, thank you. Autotrophs, they don't make their food. That is where we belong to. We depend on the autotrophs. Then based on, so we say that autotrophs, they use carbon to make their food. All right, so that's what we mean by classification based on carbon. Then the heterotrophs, they don't use carbon dioxide. We breathe carbon dioxide out. We don't use it, right? Two groups of organisms based on the source of energy. So for organism that takes energy from chemical, which is where we belong to in code, we call it chemotrophs. You get your energy, you get it from chemical source. But organisms that get their energy from light, sunlight, photo, we call them phototrophs. All right. Now, so that's what you have here splitted into that hover. If you're looking at the carbon source, the ones that use carbon dioxide as their source of nutrient, source of energy, as their source of production, rather, we call them photoautotrophs. They use carbon dioxide, they also use sunlight, so photoautotrophs. Now, if they use chemical compound and also carbon dioxide, we call them chemoautotrophs. So the difference between chemoautotroph and photoautotroph is that photoautotrophs use sunlight, chemoautotrophs do not use sunlight. All right. Now, the other portions are the other ones rather are the photoheterotrophs. Again, they are not producers. Autotrophs are producers. The photoautotrophs, they are consumers, but they use energy. They use sunlight as their source of energy. So we call them photoheterotrophs. The one that does not use sunlight, that they depend on chemicals alone, call them chemoheterotrophs. All right. So those, the chemoheterotroph, you see some bacteria there, you see some archaea there. Then for the photoautotrophs, you have bacteria as well. Photoheterotrophs, you have bacteria as well. You have some purple green bacteria. Then the photoautotrophs, of course, you see the sulfur bacteria there. Of course, you always see anything green there. So you see you have green sulfur bacteria. That's one of the things you want to take note out of them. You always see green when it comes to the anything photo. All right, let's go. Now, based on nutrients, chemical and energy requirements, organisms that use organic source for their strength, for their energy, we call them organotrophs. Lithotrophs, they don't use organic compound, they use inorganic molecules. So organisms that depend on organic molecules, organotrophs, inorganic molecules, lithotrophs. All right, now, based on their oxygen requirements, some organisms require oxygen, some don't require oxygen, some will die in the presence of oxygen. So in the presence of oxygen, some are going to survive. That's what they need. They cannot survive in the absence of oxygen. Some organisms cannot survive in the presence of oxygen, and some can do either ways. Now, anytime you say the word obligate, it means it is important, it is compulsory, it's an obligation. When you see the word facultative, it means they can do either or. All right. So when you see obligate, no, when you see aerobes, for instance, now, aerobes, they means that those organisms are usually they need oxygen, aerobes. When you see anaerobes, it means they are going to thrive in the absence of oxygen. They don't need oxygen to survive. But when you now see obligate there, obligate means the only point where they can survive is the presence of oxygen. So when you say obligate aerobes, it means they can only survive in the presence of oxygen. When you say obligate anaerobes, it means they can only survive in the absence of oxygen. So that means if oxygen is absent, they cannot survive because they are obligate non-oxygen adapting organism. But obligate aerobes means they can only survive also in the presence of oxygen. Of course, when you think about the obligate anaerobes, you may be wondering, how is that possible? Of course, oxygen, while it is good, it can also be toxic for some organisms, even for us. So the oxygen, depending on the derivative of the oxygen, they could be toxic for us. Like there are some organisms now that we call reactive oxygen species. We call them free radicals. They are dangerous for the body. They can cause mutation and lead to a problem. All right. So these are toxic oxygen, those four of them: singlet oxygen, superoxide, peroxide, like for instance, hydroxyl radicals. Those are toxic oxygen. Now, many organisms live in various oxygen concentrations. So aerobes, for instance, they are able to live in locations where you have oxygen. Then anaerobes, they don't need oxygen. All right. Now, facultative anaerobes. Now, for them, either there's oxygen or not, they can grow. They are good. Either or, they are good. Now, the only thing is that it's just that they are going to adapt to the environment. They adapt to the environment at any point. They prefer to live in the presence of oxygen, but if there's no oxygen, they're just going to change to fermentation instead of doing proper cellular respiration, and then still be okay. That's what facultative anaerobe do. Now, for aerotolerant, key thing about them, you can see the word, it says tolerant. So it's more like they tolerate, they are tolerating. Now, they don't use oxygen by default. All right. So they don't use oxygen. What they do is that they can protect themselves when there are oxygen available, because they are not able to thrive ideally. Excuse me. So, air tolerant, they don't use oxygen, but if you have those reactive oxygen, they are going to tolerate it, but they are not going to thrive in the presence of oxygen. So they are able to get themselves up, so they can. That's what they do. Micro aerophile, like the name implies, they just need little amount of energy of oxygen. Micro aerophiles, so they need very little amount of oxygen. So some of them here. Again, obligate aerobes, they need oxygen. They can only, it's a requirement for them. Obligate anaerobe, it's also required that there's no oxygen. They don't need oxygen. If oxygen is available, it will be toxic to them. So they don't need it. All right. Facultative aerobes, they could do well with oxygen, but they can also do without oxygen. Like I said, they are going to be more active in the presence of oxygen. They are more metabolism is better in the presence of oxygen. Well, when oxygen is not there, they are still fine. Then you have the aerotolerance. These we say that they are unaffected by oxygen levels, so they can tolerate. It's more like they can tolerate the process. They are good with, and then they are able to tolerate either ways. That's key thing for them. So we say that they don't use oxygen, but if oxygen is available, they will still be okay. They tolerate the whole thing. The major reason we use the word tolerant for them is because they have a mechanism to protect themselves even from the oxygen that could be toxic, so they can tolerate them. All right, now nutrient. That's part of the chapter 5, so I'll be talking about them alongside in a way. Now, in photosynthesis, there is something we have a carbon cycle, and carbon cycle there is reduction, there is carbon fixation, and there's regeneration. Those are the three things that happen in the carbon cycle. Carbon cycle is actually the place where carbohydrate is produced, right? And it has three stages. It has the reduction phase, that's carbon fixation, and then it has regeneration. The reduction is just going to be that the the substance is receiving an electron, which is what the concept of reduction is. And then the carbon fixation is a point where plants stores the carbon from the carbon dioxide that they breathe in, and then they use it to make ATP, to make glucose, to make carbohydrates. So what you call carbon fixation in over there is how plants stores the carbon. So nitrogen fixation also means that there are some bacteria that help to fix nitrogen. All right, they fix nitrogen in the atmosphere. So that's why you talk about atmospheric nitrogen, all of that. Now, so anabolism often ceases due to insufficient nitrogen. All right. So sometimes, because if you look at it critically, sometimes the body cannot do some anabolic process, all because nitrogen is absent. Think about it. Nitrogen is present in your protein. So if there's no protein, there can't be if there's no nitrogen, you can't build protein. Nitrogen is present in RNA, in DNA, is present in all of these nucleotide as well. So if nitrogen is not present, then you are not going to be able to synthesize those organisms, those substances. So nitrogen, that's what we mean by anabolism could cease due to insufficient nitrogen. Nitrogen acquired from organic and inorganic nutrients. All cells recycle nitrogen for amino acid and nucleotides to be made. Those are very essential. Nitrogen can help, by certain bacteria is essential to life on Earth. So nitrogen is very important. Imagine that there's nitrogen in amino acid. So that means if there's no nitrogen, there can't be protein. That's the logic. No nitrogen, no protein. And of course, no nitrogen, no nucleic acid. Nucleotide is the basis for DNA and RNA. All right. All right. So I think, all right. So phosphorus is also very important. You see phosphorus in DNA as well. All right. So phosphorus is important as well. You see it in DNA, you see it in RNA, you see it in your cell membrane. All right. So those are very important. Sulfur as well, very key. You see it in some amino acid also. Of course, cysteine have amino acid sulfur there. There are some substances we call trace elements, and the chief of them is iodine. For instance, now if you have iodine, if you don't have iodine or there's deficiency of iodine, that's what leads to goiter. It affects the function of the thyroid gland, and that will very affect the cause goiter. It could cause goiter. All right. Now, okay, the other ones there are the growth factors. They are necessary organic molecules that cannot be synthesized by the body by certain organisms. So they need to depend on that, on other sources rather. Those are some of the growth factors that I just mentioned now. Amino acid, of course, is part of them. Cholesterol is part of them. Heme is part of them. Of course, heme is the hemoglobin that you see in blood. Hemoglobin, heme. Now, the heme is the one that carries the oxygen there, in there. So the hemoglobin that we talk about is formed by heme, Heme, and globulin. Globulin is a protein, all right? So the heme is a growth factor. Then you have NADH, which is the one that has accepted the electron, all right? And I don't know if I got there last week, but one of the key things you want to know is that NADH, they are called electron carriers, and then they are able to carry electrons to the electron transport chain. At the electron transport chain, one NADH can make equivalently 3 ATP, equivalently. Then you have niacin, then which is the precursor that produces NADH, and also you have para-aminobenzoic acid (PABA), which are used. It helps in the production of folic acid. So in the transition of folic acid, it's an intermediate structure in between. I think that's it. So take note of the growth factors as well. Okay, now on the requirements that organisms need for survival, physical requirements. Now, temperature is a chief factor. pH is another one. So for instance, now you notice if you have been in the lab, which I believe some of you have, you notice that whenever you want to do, when you're going to do an experiment, or whenever you're going to culture any cell, you will notice that you will be asked to put it in an incubator. Now, in the incubator, the incubator is about 37 degrees Celsius. So in reality, it's about it's about the normal room temperature. It's about that. So that's why you notice that whenever you have bacterial infection, you have fever. Why do you think you have fever? Or why do you think you have fever when you have bacterial infection? Can you guys hear me?
Yes.
Is that an indicator that people are not with me?
Yes, we are hearing you, sir.
Okay. So answer my question.
Yes, we can hear you. We can understand your question, but just we don't know the answer.
Hey, no, I don't mind that you don't know the answer. I just mind if I don't get response.
Yeah. Maybe
I
at that time.
Okay. So someone said the fever fight the bacteria. Okay. What else? Any thoughts? I'm not asking for perfect answer. I just want your responses on it. It's just use
The
logic. It
fever
doesn't
is
have to be
a
accurate.
response from, it's an inflammation response.
Okay, cool. Now, what fever does, it's of course immune response, like you guys said. That's it. That's just it. But what actually happens is this: whenever you have fever, fever is out, is our defense mechanism. Yes, that's it. So what happens is when you have bacterial infection, for instance, your immune system detects that there's a stranger here. Now, when the immune system detects a stranger, the immune system raises an alert. Now, what that does to the body is that it automatically tells the body that, okay, there's a foreigner here. Then the immune system make a scan and check the texture of what kind of bacteria, what kind of organism is this foreigner. So then that helps to know what to do and how to attack it. So what the reason you have fever is this. The bacteria will be able to survive well at the same room temperature, the same temperature in your system. So the purpose of fever is to raise a temperature that is higher than the temperature where the bacteria can survive. So usually what it does for some bacteria is that it's going to stop them from being able to make protein because they are not going to be able to do that at that high temperature. And then it makes them to be dormant and not be able to do their activities, which now give your body the chance to fight them. So that's what some of the bacteria does. Yeah, so that's majorly what they do. So the temperature is the purpose of fever is actually to raise a temperature that is not conducive for the bacteria to survive in. Right? That's it. Now, immediately that happens, your body secretes a chemical. It's called pyrogen. So it's a pyrogen that raises your body temperature. And so that makes the environment to be too hot for the bacteria, and it could affect their protein synthesis. Right? Please give me a moment. Just excuse me a little bit. So, however, so the optimum temperature that has been discovered for bacteria, for most of them, is about the temperature in our body. And that's why you see that we put them in the incubator, which is also about that temperature. Whenever you want to do any experiment, you put the bacteria in the incubator and let it go for 24 hours. So when the bacteria also gets in our body, they are comfortable with the temperature there. So they can usually just replicate and produce their protein. When they produce their protein, that's what leads to infection. But if the protein is there, if the bacteria is there, but they cannot produce protein, then they cannot hurt you. So a bacteria can be in your system and be dormant. There's some of our antibiotics that you take, some of them, they are bactericidal, some of them are bacteriostatic. Bacteriostatic does not kill the bacteria. Antibiotics that are bacteriostatic doesn't kill the bacteria. They just make the bacteria to become static. They are going to be in your system, but they cannot hurt you. Because when they cannot produce protein, just the same way if you have a gene and that gene cannot produce protein, the gene has no effect on your body. It's not going to have any significance on your phenotype. That is the same way it is as well. That if you have a bacteria in your system and the bacteria cannot work, cannot produce protein, then they're not going to hurt you. They're going to be there. So bacteriostatic antibiotics, it makes the bacteria to be there, but they are static. They can't hurt you, but they are not dead. What the advantage of that is this: when you take such antibiotics, your body now develop an antibody against that bacteria. Then your body fight it by itself and kill it by itself because it's there now as a soldier that cannot hurt you. So your body develop antibodies and kill it. But you are not going to give that kind of antibiotics to someone whose immune system is compromised. Maybe someone who have prior issues of health like diabetes, high blood pressure, people whose immune system is compromised. You're not going to give that to them, because if you give them that, their body cannot fight the bacteria. But if you give someone whose immune system is active, then the immune system will be able to fight the bacteria by itself. So for people who are immunocompromised, you give them bactericidal so that the antibiotics can kill the bacteria. All right. So, however, as well, there are some temperature that is the optimum temperature. Optimum temperature means the temperature where the organism is at its best. When I say at its best, it means that it's performing the best at that time. it's doing its protein replication, protein production at the best rate at that point. That's what we call optimum temperature. It's the same thing. Optimum pH, optimum is the temperature or the factor where the organism is at its best. Now, listen, look at this. Look at this. Optimum does not mean minimum. All right. So optimum does not mean, it doesn't mean minimum. It doesn't mean maximum. It means the temperature that gives the best result. This here is the optimum. All right. Now, it is not the middle. It is not the middle. No. Usually, it is usually above the middle, between the middle and the maximum. But it's not the middle, it's not the maximum. It's an entirely different parameter. All right. So if you look at this now, which of these beaker do you think, which of these dish do you think shows the optimum temperature? Which of these three? Is it the 22 degrees Celsius, the 30 degrees Celsius, or the one that has the 37 degrees Celsius? Which of them do you think shows the optimum?
37 degrees Celsius.
What did you say?
37 degrees Celsius.
All right. Yeah, thank you. So you see the 37 seems to be more, to have more result. Yeah, you see more colonies there. 22, so this is not good. This is good, but the 37 gives about the best temperature there. All right, let's go. Right, now this here shows different organisms and different factors, looking at different expressions that you can see in organisms. So you see hyperthermophiles means that they are able to survive high temperature. Very high temperature. This is thermophile. This is high temperature. Thermophiles, high temperature. This is very high temperature. It's beyond the normal. All right. So you see them between 67 thereabout up to almost 110 or 105. Those are hyperthermophiles. Now, you see some organisms are able to survive here between here and this range, early 40s, like 43 up to 80. Those are thermophiles. We call them thermophiles. Now, some organisms are able to survive from here to here. We call them mesophiles. And the one between 0 up to 40, we call them cycrotolerant. And then the one that are between -4, 5, thereabout to 20, we call those psychophiles. Now, psychophiles means that they love cold. All right, they are able to survive in the cold, and of course, you can tell with the temperature. This is less than 20 degrees, so we say that they are psychophiles. So they thrive in temperatures. Usually, we say in the average, you always see them thriving in temperatures lower than 15 degrees. All right, but if still up to 20 degrees, we still call them thermophiles. Psychophiles. Okay. Psychotolerant means that they are able to, of course, the word tolerant, they are able to tolerate those temperatures within that range. So they are still, all of them are extremophiles. All organisms that live around these temperatures here, we call them extremophiles. So they thrive in low temperature as well. Key thing. Let me see if there's any other thing. All right. Now for pH, still the same concept. There's going to be optimum pH, which is a pH that you see that the organism is thriving very well. Organisms are sensitive to changes in acidity. pH deals with acidity, right? Or alkalinity. So if it is between 0 to 7, we say alkaline, acidic. Between 7 above 7 up to 14 is alkaline. So neutrophils are organisms that are within the medium, within 7. 7 is the middle of that chart. So they do well at the neutral pH, which is neutral pH is of course 7. All right. Acidophiles, they grow better in acidic environment. So you see them thriving any pH below 7 degrees. All right. 7 pH of 7. Then alkalinophiles, you see them thriving anything above 7 degrees up to 14. Those are alkalinophiles. Right? So generally, we use the word neutrophils for neutral, alkalinophiles for the ones that like basic, then acidophiles for the ones that are surviving in acidic environments. Right? Now, the allophiles, I don't know, I don't think if I will still see it, but allophiles, I believe I should have it somewhere later. I just want to point to it now. Allophiles are the ones, excuse me, I don't know why this thing like that. Just wait. So allophiles, okay. Allophiles are the ones that they like salt. Sorry, halo is HALO. All right. So allophiles are the ones that like they are able to survive in salt environment. Allophiles, salt. So they can survive up to like 30 degrees salt, 30% salt environment. They are still good there. Of course, you know that our own salt environment is about 0.85, not even up to 1. So it goes between 0.85 and 0.9. But these allophiles, they can do up to 30%, up to 30% NaCl. So we call them allophiles. Now, physical effect of water, of course, this will be like a refresher for some of you now. Now, microbes require water to dissolve enzymes and nutrients. However, just like I was talking about halophiles now, their water is always of different degrees. Like for us, our environment is like 0.85 NaCl, meaning that would be like 91 point something water, right? So most cells die in the absence of water. Water is necessary for metabolic activity. And of course, too much water can cause a problem as well. So we talk about osmotic pressure and hydrostatic pressure. So I will discuss, like when you talk about osmosis, you are talking about how water moves. When you're talking about hydrostatic pressure, you are looking at how like the blood against the artery, for instance. That cross-border process, that is what we talk about right now. Sometimes when bacteria, for instance, they are able to form what we call endospores. They form endospores particularly when they get to unfavorable condition and they are looking for ways to survive. So it's more like they are closing down. Just the same way some of us, when you get to environment, when you know that you are not comfortable there, you just shut down. So bacteria also are able to do that. They shut down. Now, when they shut down, they are not dead. But what it means is that because they are shutting down, their activity reduces. That means their demand also reduces. Metabolic demand reduces because their metabolism is also going to reduce. So they end up not needing resources to survive at that time. So we say that they form endospore at such point. It's something you will hear very well, particularly in the lab as well. Now, when we talk about movement of water, which is the osmotic pressure, a solution. So we have a solution that we call hypertonic. Okay, this is allophouse here. So we have a solution that we call hypotonic. Hypotonic means it has more water. So you see, it's more a solution that have lower solute concentration, meaning that it's going to have more H2O. So when you place a cell in this hypotonic solution, what's going to happen to the cell? So think about it. This cell have less H2O, whereas the solution have more H2O. So what's going to happen? Water in osmosis, water move from where water is high to where water is less. So that means water will move from this environment into the cell. So what's going to happen? The cell will swell. When the cell swells, it gets to the point when after swelling, it then may burst if it is a red blood cell. And when it bursts, we call that hemolysis. However, if you put a plant cell in this environment, the game is different. For plant cell, they won't burst because they have cell wall. They also have central vacuole. The central vacuole makes them, they use the central vacuole to store water, and then the cell wall is cellulose, and it doesn't make them burst. Hi, baby. Sorry. So it makes them to, they use the central vacuole to retain water, and they use their cell wall. Their cell wall have cellulose, which is a polysaccharide that is tough. It doesn't make them break. All right. Now, that's what happened for plants. So all that happened in plants, hemolysis doesn't happen in plants, or hemolysis. What happened in plants will be turgid pressure could increase, but they are not going to burst. Now, hypertonic solution have greater solute concentration. So what does that mean? It means the solution will have less water. So less water will be here, whereas more water is inside the cell. So more water inside the cell. So what does that mean? Water will move from the cell into the environment. So that means the cell will lose water. The cell loses water. So when the cell loses water, then what happens? The cell shrinks, shrivel, and we call that crenation. So environment, there are some organisms that are able to live in salt environment. We call that allophouse. Now, allophouse could be obligate, meaning that they can only survive salt environment, or they could be facultative, which means they can switch in between the two, either salt or water environment. All right, those are the organisms there. Let's go. Barophouse. Anywhere you hear baro, it is pressure. Pressure. So some organisms, they are able to live in very severe pressure. We call them barophouse. All right. Then association and biofilms. Now, I told you already what biofilms are. Biofilms is when bacteria make seal surfaces, like forming plaque. Very good. That's the way to think about it. Is biofilm is the best way to think about biofilm is think about it like plaque forming on your tooth. That is biofilm. It is. So it's bacteria coating surfaces. It's like they are laminating the surface. All right. And of course, biofilm is made up of bacteria, and there is matrix, which is what seal up the bacteria on the surface. All right. So we have some antagonistic relationships when the organisms are against. They are not in synergy. We have synergistic. Synergistic means that they are able to produce amplified results. Amplified results means it's not just 1 + 1 = 2, it's 1 + 1 could equal 5. We call it 5. We'll still talk about things that are relating to that. Then symbiotic, of course, is mutual benefit. All right. So biofilm, it's a complex relationship that you see among numerous microorganisms, and you see them on surfaces, like I said. So you're going to see them on surfaces. You could see them on some devices because they built up there and not sterilized or cleaned over time. You could see them on mucous membrane as they build on the digestive system as well. So they could be there and then they build over time, thicken and glue to the wall of the digestive tract as well. Now, many of microorganisms are more harmful as part of a biofilm. So when they accumulate like that, which is what I was pointing out there, if they do the synergistic effect, then they are able to overweigh the body than individual efforts. All right. So this is what biofilm looks like. This just exactly what I am trying to point out to you. So you see, this is like bacteria forming on this area, and then they are being sealed into the space, more like laminated. That is biofilm. Okay. Now, if you are already in the lab, you should be hearing or saying the word inoculation already.